And the next one, the statement 1, the glycolytic pathway is the only route available for the oxidation of sugar in plant cells. And the statement 2, in pentose pathway, the complete oxidation of one glucose 6-phosphate molecule to CO results in the production of 12 NADPH molecules. So we need to analyze the two statements and identify it. First, let us take the first statement. So glycolytic pathway is the only route available for the oxidation of sugar. No, it is not a only pathway. It is a double way pathway. HMP pathway is also there for oxidizing the sugar. So the statement 1 is wrong. Okay. And the statement 2, so pentose pathway is a complete oxidation of glucose 6-phosphate, which will be results in the 12 NADPH molecule. So this statement is correct. So the statement 1 is incorrect and the statement 2 is, statement 1 is incorrect and statement 2 is correct. So the option D will be the correct answer. Okay. And the next we will move on to the next question. Statement 1, telomeres or the sequence at the end of eukaryotic chromosome, which helps in stabilizing the chromosome. And the statement 2, telomeres end with a multiple repeats sequence in the form of TTAGG in humans. So we need to identify which statement is correct. Okay. So from this question, both the statement is actually a correct statement. Okay. The option A is correct. The telomere, which is at the end of the chromosome, will help to stabilize the chromosome. As well as it will, at the end, it will have a multiple repeats like TTAGG in the humans. Okay. So both the statements are correct statement. Okay. And the next one, statement 1, in the agarose gel electrophoresis, the large pore size of low concentration gel makes it possible the separation of very large molecules. And the statement 2, the agarose gel electrophoresis, the small pore size of high concentration of gel makes a possible separation of large molecules. Okay, so for this question, the option C will be the correct answer. Okay, so in the first statement, they have given that, so the large pore size of low concentration gel will be separating a large molecules. It's correct. Okay, so because when we are putting a low concentration of gel, what happened? The pore size will be very small so that the large molecules will be deposited at the top of the gel itself. Okay, so we can easily identify the small fragments. Okay, this statement is actually a correct statement. And the statement 2, the small pore size of high concentration of gel is possible of separating a large molecule. If the concentration of gel was high, what happened? The pore size will also high. It cannot able to separate the large molecules. Okay, so the statement 2 is wrong. So the option C will be the correct answer. Okay, and the next one, the statement 1, the mycorrhiza is a symbiotic association between the cyanobacteria with the roots of higher plants. And the statement 2, the mycorrhizal association is important for nutrient acquisition in the plants under the limited condition. So for this question, the option D will be the correct answer. Okay, so the first one, the mycorrhiza is a symbiotic association between the cyanobacteria with the roots of higher plants. Okay, it is actually a wrong statement. Okay, so the mycorrhiza is a fungi which will go for the symbiotic relationship between the plants. Okay, and the statement 2, the mycorrhizal association is important for nutrient acquisition in plants under the limited condition. Okay, so as I already told, it will be useful for the nutritional benefit. It will go for the symbiotic relationship. It will transfer the nutrition to the plants as well as the plant will transfer the nutrition to the fungi. Okay, so at the certain condition, both will get benefited. So the statement 2 is correct. The statement 1 is wrong. The option D is the correct answer. Okay. And the statement 1, apoptosis is genetically regulated process occurring in a specific tissue during a development and the disease by which cell destroy itself. And the statement 2, apoptosis is regulated by intercellular signals generated from many secreted cells, cell surface, protein hormones, as well as the many environmental stresses. Okay, so both the statements are actually a correct statement. Okay, so the apoptosis is a genetically regulated process and it will be helpful for the specific tissue for the development and it can able to destroy itself. Okay, and it is regulated by the intercellular signals. Okay, and it will be receiving the signals from different kind of cell surface or different kind of protein hormones, or due to the environmental stress, it will receive the signals. Okay, so both the statements are correct. So the option A will be the correct answer. Okay, and the next statement, in C4 cycle fixation of carbon dioxide by carboxylation of phosphoenol pyruvate in the mesophyll cells results in the conversion to malate and or aspartate. The statement 2, the transport of C4 acids to bundle sheath, that leads to decarbylation and the generation of CO2, which is finally reduced into carbohydrates via Calvin cycle. Okay, so for this question, the option A will be the correct answer because both the statements are correct. Okay, so for C4 cycle fixation, the carboxylation of phosphoenol pyruvate will be happening in the mesophyll cells. Okay, it will convert the malate to aspartate. Okay, as well as that C4, there will be a transport of C4 acids will be transported to the bundle sheath. So while transferring, it will form a decarboxylation and there will be a release of CO2 molecules. Okay, so there will be a reduction of carbohydrate group glucose molecules will be reduced. So this process will be happening with the help of Calvin cycles. Okay, so both the statement 1 and 2 are correct. And the establishment of mammalian cell culture involves following steps. Option A, culture of primary cells, subculture of primary cells, separation of cells from the tissue by mechanical or enzymatic method, culture of secondary cells, subculture of secondary cells. Okay, so we need to arrange in the order of the establishment of mammal cell culture in the following steps. Okay, so the option A will be the correct answer. So first we need to separate the cell from the tissue or organ. After the separating, we need to give the mechanical description for the release of cells. Once we have isolated from the tissue, from the mammals, or if it is an animals, whatever. So after that, we need to go for the mechanical disruption or by the enzymatic action where it will release the cells, where it will release the target cells. Okay, once it releases, we need to go for the primary cell culture. Okay, so for adapting the condition for the where the primary cells will have a same kind of nature as the original cells will have. Okay, so after the growth of primary cell culture, we need to go for the subculturing process to check the stability, to check the how long the cell is prolonged, how long it will be sustained. For that, the subculturing of primary cells need to be done. Okay, and after primary cell culture is after the subculturing of primary cells is passed, next we will move on to the secondary cell culture. Okay, secondary cells, for it will also have the cell lines where it will have finite cell growth and or continuous cell growth. Okay, so once the secondary cell culture is over, we need to go for again the subculturing technique of secondary cells. Okay, so the order will be C, A, B, D, and E. Okay, the option A will be the correct answer. Okay, and the next question, the steps involved in the bacterial translation are. So what is translation? So it is the conversion of mRNA to the protein molecule. Okay, so we are going to arrange the steps for the bacterial translation. Okay, the first we activate the amino acid. Okay, the activation of amino acid will be the first step, and there will be a formation. The amino acid will convert into amino acid tRNA. Okay, so this is the second step. After amino acid formation, there will be initiation of polypeptide synthesis. Now amino acid is formed. We need to connect the amino acid by the peptide one. Okay, so once peptide is formed, it will need to form a n number of amino acids so that it can make a complete protein. Okay, so the polypeptide is now synthesizing, initiation of polypeptide synthesis. After that, the peptide bond formation. Okay, and the last one is termination of polypeptide. So once the process is going to be end, we need to cut the remaining bonds. Okay, so the option A will be the correct answer. A, C, B, D, and E. Okay, and the next one, the sequential steps involved in the prophase 1 of meiosis are. So for this, we need to arrange the step. Okay, the option A, chromosome condensation, terminalization of gemsta, formation of tetrad of bivalence, pairing of homologous chromosome, genetic recombination. We need to arrange in the order. Okay. So for the prophase 1 of meiosis, the first thing will be chromosome condensation. Once it condenses, it will form a pairing of homologous chromosome. Okay. Next one is D, pairing of homologous chromosome. And after that, there will be a formation of tetravalence. 4 strand will be formed. Okay. So next, genetic recombination will be done, and the final process will be terminalization. Okay. The end of chiasm. Okay. So the option C will be the correct answer.